Hello everyone, good morning or good afternoon. I'm Nika Estiri, and my title of talk today is Decoding Aging Mechanism Using Synthetic Minimal Model. This project has been motivated by Tae-Suk Moon, John Glass, and myself here at J3I. And um, today I'm excited to share our vision of how minimal cell can help us to understand the basic mechanism of aging and ask questions about aging. But before going forward, why aging? Aging, one of the biggest risk factors for disease like Alzheimer, cardiovascular disease, and cancer. If we understand the basic mechanism of the aging, we are able not only extend the life, but importantly, we can extend the health span. So maybe one of the most important questions today in biology, or more exciting question, is that how we can slow down aging, prevent aging, or even reverse aspect of the aging. But before answer to this question, we need to define aging. In human or in eukaryotic cell in overall, biological aging is defined by many things, including but not limited to telomere shortening, mitochondrial decline, stem cell exhaustion, epigenetic changes, and so on. And it is interesting that aging is not an issue for the human. It's even plants can experience the aging. But the other thing is that what about other organisms like microbes or bacteria that doesn't have a tissue or organ? What about them? Do the bacteria age too? So maybe think for a minute. If a bacterium divide, is one cell younger than the other? Does a bacterial mother cell actually exist? Can a microbe die of old age, not just starvation or stress? Or even could a minimal cell with less than 500 genes also show sign of aging? For for a while, for many decades, our assumption was that binary fission is equal to perfect rejuvenation. When a bacterial cell starts to divide, it will create two identical cells that are 100% the same. So in this case, it can lead to the unlimited division. But it turns out around 2 decades ago that this assumption actually is not no longer true. The first evidence came in 2003 when scientists showed that there is a bacteria called Colobacter crescentus. This bacteria obviously and naturally shows asymmetric division. During the cell cycle, it produces a stock cell, a mother cell that stick to the surface and accumulate damage and will senescence. On the other hand, a swarmer cell, a daughter cell that has a flagellum and it will rejuvenate. So this was the first clear evidence that shows that the bacteria actually senescence. But what about other bacteria like E. coli that it seems that divide symmetrically? In 2005, Stewart and others showed that even in this bacteria like E. coli, there is a two pool, old pool and new pool inside the cell. The cells that receive the old pool actually they divide slowly, they grow slowly, and they produce less offspring and die more often. So even the division is seems that is symmetrically, but functionally and physiologically is completely asymmetric. This was the evidence that showed that the idea of that the bacteria is immortal actually shattered from 2005. But to prove this, we needed new tools to show or track individual cell among many generations. And this was the time that mother machine came in. A mother machine is a microfluidic device that can trap a single mother cell in the closed end of the tiny channel. Here there is a movie of a mother machine that shows how it works. It shows in the bottom actually they are the closed part that the mother cell are trapped. When the mother cell divide, it actually it actually produces daughter cell that this daughter cell washed away to the other side that is open, and the the other side during the washing with the fresh media, the daughter cell completely will wash out. This technology allows scientists to actually track a same mother cell for several hundred generations, and this technology showed that bacteria actually do age. So this brings us to the Scene 3.0, the simplest self-replicating cell with only 473 genes. As John mentioned, from this number of 433 genes, actually 400 genes are essential that we don't know the function of 66 of them, that is around less than 17% of the genes. Also, although there is the autolog of these genes is almost all level of the life. But among these 66 genes, 35 of them are completely unknown, and there is no idea what they are doing. And the rest of 31 genes are called generic. We know they are a hydrolase or a transporter, but we don't know really the substrate or and the reaction purpose. Our idea is that maybe some of these essential protein of unknown function may actually stay in the mother cell in the old pool and maybe factors the cell aging and tie to aging. So for testing our hypothesis, we want to use minimal cell. Minimal cell is a uniquely simplified platform that's ideal for investigating fundamental question in biology, especially biology of cellular aging. So our idea is to attach the minimal cell that it can be with several methods. One of them, it can be just using biotin tag that has a strong affinity to bind to a streptavidin coated surface. This allows us to attach the mother cell and can have a separation from the daughter cell that will be washed out during the with the fresh media inside the system, and this can be incorporated inside the microfluidic chamber like the mother cell that will allows us to track the minimal cell for maybe several hundred generations to see how aging works in our minimal cell. So, of course, the data alone is not enough. We need to integrate multi-omics. We need to profile these cells to see how they will go to during the aging. For this, we will look at the data like genomics. We will look at the mutation of the genome during the aging. We will collect the data for transcriptomics to see how gene expression will drift during the senescence. We also look at the metabolomics and look at for the byproducts or toxic compound that may be accumulated during the aging. And also we will look at the proteomics for misfolding or aggregation of the protein, especially this essential protein with unknown function that, as John mentioned, maybe this non-essential gene maybe actually are involved in the aging, and we will look and profile this protein to look at how they are stay or they are in the old pool of the cell that can be accumulated during the aging in the mother cell. So by looking at in the different layer of the information, we can have a holistic view of the aging and to see how a minimal cell age during many generations. Of course, data alone is not enough. We need a computational model. We will integrate our multi-omics data into computational model to simulate and predict the trajectory of aging. And our workflow for this, including data collection, model building, and insight generation. With this, our minimal cell from a descriptive system and model will transform and convert to a predictive model. Our broader vision is that we hope to uncover universal aging mechanism. This insight will help us to uncover the conserved and common pathway that is conserved during evolution or during the different organisms. This can lead to the design of targeted intervention that can lead to the extend healthy lifespan. And also, this has the potential to build a scalable research platform that can be rapid, cost-effective, and high throughput that allow for a screening anti-aging compound. So to summarize, aging is not a problem just for human, bacteria age too. And we hope that with minimal cell, it can uncover many of the fundamental questions, especially aging and cellular biology of aging. With that, I want to say thank you for your listening, and I'm happy to answer your question.
Okay, thanks for your great talk. I just have one question. How many cell generations are you going to cover with this experimental protocol?
I don't know exactly, but this is we expect at least for more than 100 generations.
Okay, cool.
Dan?
I was just say, Engwan asked a very good question because he's doing the computational study of multi-generations of using the model that we have. We can't do it fully spatially because it would take too long. It takes 4 to 5 days just to do 50 replicates. But what we're trying to do it is with the, the more, how should I say, where you don't have the spatial diversity, a model of it. And we do see differences in the cells already in terms of how much surface area, how it changes, also other things that come in. So he might talk to you and just show you his preliminary results because we haven't gone over them enough for him really to present. But it gives maybe some interesting things. And I forgot how many generations you've done, Engwan. How many have you done?
5 in total.
We're a little behind. But it's already interesting, particularly those ones that in Syn3A seem to, there's still a few that are non-essential in Syn3A, and they appear in varying amounts, but some of them get less and less and less and less and just disappear altogether, the proteins from them. But anyhow, no, great talk, beautiful work.
Thank you for your comment. Thank you. Definitely, we will contact all the people that's working on the microfluidic or any experience in this field, computational. We will work on it and we will contact you definitely.
So Zan and Engwan, Tae-Suk Moon, who I don't think is with us right now, has acquired funding for this project. So we are expecting to start this. Nika will actively begin doing experiments in 2026, I hope.
Cool. We can make already some suggestions. One would be shift to Syn3A, but okay.
and then Kim, you're up. Last question.
You're mute, Kim.
My apologies. Questions about mutations and offspring generations. So we know it mutates extremely quickly because of error-prone stuff. So if you have one cell sort of stuck on the grid or on the template, is it going to be correct that the strand that the DNA strand that gets that divides, will the new strand, that's the one that will have mutations, will it be the one that is shed, and will the original just be always free of any mutations? And will this affect, your downstream populations?
Yeah, but I think we will need to look at the different generation and especially young versus the mother cell or the old or even the old maybe different layer. I'm not sure how it exactly will work or how we need to completely separate, the mother cell that is single, for example, for many generation. We need to really look at it, but that's a great consideration.
Okay.